Good afternoon. My name is Jane Bai, and I would like to welcome you to section 2. In section 2, we will focus on overview of QSP and successful uses of QSP in drug development. We have 3 outstanding speakers: Dr. Stephan Smith from University of Florida, Dr. Brian Smith from Bristol Myers Squibb, and Dr. Susana Zaph from Sanofi. And their presentation will be followed by a panel discussion, and Dr. Justin Earp will lead the panel discussion. He is a team leader in divisional pharmacometrics, and he is also a co-chair of System Pharmacology Scientific Interest Group. And now I would like to invite Dr. Stephan Smith to begin his presentation.
So thank you very much. First of all, before I start, I would like to thank the organizers for having me speak this afternoon, specifically Dr. Spey and Earp for their kindness, also of having me here at FDA. All right, so thank you for the help here. Apologies for the delay. So I also like to thank Drs. Lesko and Rogi for their input on my presentation. So 3 things I would like to accomplish today. Number 1, I would like to give a brief overview of the history and current state of MIDD approaches and their use in drug development and regulatory evaluation. And Dr. Sine had briefly alluded to that earlier today, so I will keep this very short. Second, I would like to give a perspective on current barriers to routine application of QSP model in drug development and regulatory evaluation. And please take this as my personal perspective. And then third, provide a perspective on what I personally think we should do next. So as Dr. Sine alluded to earlier today, modeling and simulation and MIDD approaches have been around for a while, initially starting with mainstream applications in the 1980s with Lou Scheiner and Stuart Beal. And you see that the terminology has evolved over time. So the term pharmacometrics and systems pharmacology came up relatively early on in the 1990s. But I also want to point your attention to the fact that there were a number of key publications as well as key initiatives at the US Food and Drug Administration, such as the FDAMA in 1997 that really laid the groundworks for model-informed drug development approaches as we know them today. Again, the term systems pharmacology appeared in the literature relatively early on, but I think we are somewhat plagued by the fact that we still have not an overarching definition of what exactly it is. Pete Hine, Fallerhav, and Neil Benson took another step at this, and I agree with that definition, to be honest, in 2011, where they defined systems pharmacology as the quantitative analysis of the dynamic interactions between drugs and a biological system to understand the behavior of the system as a whole, as opposed to the behavior of its individual constituents. As we move forward in time, we see that the terminology kept shifting to model-based drug development, and then, of course, people recognizing that drug development is not necessarily based on models, but it's rather informed by models. So the terminology shifted to model-informed drug development in the mid-2000s, and depending on which side of the Atlantic you're on, we are still using the term MIDD today on the US side, and the European side has coined an additional definition of model-informed drug discovery and development, recognizing, of course, that quantitative approaches such as systems pharmacology approaches can also be very effectively employed in early research development, and they define it as a quantitative framework for predicting and extrapolation centered on knowledge and inference generated from integrated models of compound, mechanism, and disease level data, and aimed at providing the quality, efficiency, and cost effectiveness of decision making. So as far as I'm concerned, we have quite a bit of terminology already at our hands. And I think it's really time now also maybe to settle on a piece of terminology and keep it consistent and move on from there, because otherwise I am personally afraid that we basically contribute to continuous confusion of what exactly it is that we are talking about. So I also thought a little bit about where we are in terms of value proposition versus prioritization of MIDD approaches. And I call this a 2023 snapshot because obviously this is a moving target. So when you look at benefit versus how easy it is of using this modeling approach, so benefit in terms of impacts on goals or questions from low to high, and ease of doing in terms of the time, resources, data, and buying from management. In terms of easy or difficult, I think we would all agree, and I think Dr. Sine outlined this already as well, that we routinely use population approaches, so population PK or population PKPD approaches, exposure response approaches in today's drug development and regulatory evaluation processes. I think PBPK, and I'm trying to also strike some analogies between PBPK and QSP going forward. I think it's ready for prime time, but there's still the underlying notion that is somewhat difficult to use. But I think this has clearly been moving in a high impact area, particularly when it comes to the evaluation of drug-drug interactions and special patient populations, for example. So where are we with QSP? As far as I'm concerned, QSP has an enormous potential, but I think we are not quite ready for prime time yet. Oftentimes it takes a lot of domain expertise to develop and appropriately verify the models in the context of use. But I think, and that is my firm belief, as time goes by, we will certainly also move to quadrant 1. And over time, we'll be able to use these approaches as mainstream applications. Perception matters as far as I'm concerned. And there's, I think, a couple of underlying perceptions that are out there in the community. And again, I'm trying to draw a little bit of an analogy between PBPK models and QSP models. And, from talking to my QSP colleagues, I oftentimes hear the argument, yes, they are user-friendly interfaces, but they're user-friendly black boxes, so you don't necessarily know what's going on behind the scenes. And as a hardcore model, I'm not necessarily comfortable with that because I don't know necessarily what's happening. And so there's somewhat of the notion that you have this, click here, click there, click everywhere kind of paradigm. And I think so we probably need to break through this a little bit to make sure that, models in and by itself agree on the approaches, because otherwise we will probably have a hard time convincing other folks to buy in. QSP models, on the other hand, are perceived as being structurally complex, requiring a lot of data, potentially having challenges with scaling experimental data or experimental models, or models that have been established on short-term experimental data to long-term endpoints or even then clinical outcomes. And this is just a personal observation, so take this with a grain of salt. It seems to me that everybody has a bit of their favorite model companies, and I understand why, have their own in-house models. But again, that's not necessarily helping the community as a whole. And I think we should probably do a better job on agreeing some of the models for broader context of use as we move forward. Because again, otherwise, if we can't agree amongst ourselves, then we probably have a hard time with other folks buying it. So let me go back to what the idea behind modeling is. So the original idea is, of course, that you use mathematical model at various levels of complexity to clearly define your assumptions, integrate the data that you have, and ultimately make an improved decision. So, for example, waive a clinical trial as you move forward, and Lou Scheiner and Stuart Beal coined their famous learn and confirm paradigm. But what I think is really important here to realize that learning and confirming is great, but we also want to, in a broader context, basically apply the lessons learned moving forward. That does not only pertain only to what we learned a particular compound, for a particular development program, but maybe also apply some of the lessons that we have learned for establishing exposure response or PBPK models to QSP models, because there is really no need for going through the same, and in some cases painful experiences all over again. So maybe let's compare notes across the field. And so that's what I also want to do in the next few minutes of my talk. So again, going back to China and Beal, where three key questions were defined that define the context of modeling simulation. So number one is, of course, what do we want to know? Number two, how certain do we need to be? And number three, what are we willing to assume? So when we look at, what is it that we want to know, then oftentimes there's a notion, and again, I'm coming from an academic background, and this may be a biased point of view, that if you have time and resources at your hand, then obviously you try to make it as fancy as you can, but you may or may not be able to do that in a drug development and regulatory evaluation context. So you really need to pick the tool that you're using for answering a certain question appropriately to make sure that you bring value to the table. And so I found this publication from the European Journal of Pharmaceutical Sciences from Helming et al. That I think outlined this very nicely in a sense what respective tools have been used. So systems biology models for helping target selection, PBPK models. I think their strength is also that they have the ability to make inferences about tissue distribution. QSP models, to some extent, originated from safety questions, and then obviously population approaches, either population PK or population PK/PD approaches, to make sure that we basically identify covariates and ultimately can make inferences about giving the right drug at the right time and the right dose to the right patient. So obviously, and then ultimately meta-analysis and other approaches to look at the marketing potential at the same time. And obviously, these approaches carry throughout the drug development continuum. And I want to highlight here that it's really a modeling continuum, not a modeling competition. So we should not have really the discussion, should it be a PBPK model versus a QSP model, because all of these approaches are mutually informative for one another. So how certain do we need to be? And what is the impact of variability on exposure response? And you see here this type of graphs in a number of textbooks, including the Roland textbook, where you see exposure plotted versus response, and then you see these idealistic curves. So as the concentrations go up or exposure goes up, you will then ultimately achieve a therapeutic response. And as you further increase, then concentrations, you will first run into minor toxicity or then serious adverse events. Recognize, however, that these are mean curves and don't necessarily tell you much about the underlying variability. And I found this nice graph here from Friedrich that really highlights that patients are different, not only in terms of their drug exposure, but also in their ability to respond to drug treatment. And historically, and I picked this typical sources of variability from one of the original publications of Carl Beck, the former OCP director, that lists a whole number of sources of variability in both in terms of exposure as well as response. Given that we are in a rare disease context here, I'd like to put some additional factors to consider for QSP model here on the table. And so number one is, of course, what is our uncertainty in disease pathways? Some diseases are quite well understood. For example, if they are monogenetic diseases, are quite well understood. And so we talked a little bit about the development of natural history models. So I think this is really an area where QSP modeling can help, but also for polygenic diseases where we have multiple interacting pathways with multiple interacting drug targets. Really, I think this is where QSP models can help. I think we need to pay a little bit of attention in terms of what is the target. Is it an endogenous target, so for example, a receptor or a protein, or is it an exogenous target and a list of pathogens, so viruses, fungi, and bacteria? And then ultimately see what the opportunities for QSP models are. So historically, of course, preclinical to clinical translations. So for example, mapping out complex physiologic pathways, identifying targets for drug prioritization. I think this is routinely done in oncology as far as I'm concerned. Identification of optimal combination chemotherapy, but also, of course, again, in the rare disease context, I think one of the challenges that we are facing is that we have like a hard time with having placebo controls. So this, in fact, may be an opportunity here for QSP approaches to maybe be translated in virtual twins, if you will, to see if we can use that for inference making on drug response as disease progresses. And ultimately, and I think we talked about this this morning, Dr. Sine talked about this a bit already, understanding biomarker dynamics, and then based on a biomarker dynamics form optimal clinical trial design. So ultimately, of course, it's important to integrate what we know about PK and PD, and I picked a very simple example here, voriconazole. And you may say, okay, why did you pick that example? So bear with me while I walk you through the rationale. We know, of course, for voriconazole, it's an antifungal drug that is metabolized by 2C19, has genetic, is subject to drug-drug interactions and gene-drug interactions, has no linear PK, and has a narrow therapeutic window, as shown here through the two dashed blue lines. And you see that there's quite a bit of variability, either subjects falling out of the therapeutic window on the bottom end or going above the therapeutic window on the top. And of course, you can apply pharmacogenetic approaches or traditional TDM approaches to get those subjects back into the therapeutic window. And if you wanted to, you can, of course, then stratify these subjects by clinical phenotype for 2C19, for example, normal metabolizers versus ultra-rapid metabolizers. You see, on average, they are different, but they have largely overlapping variance bands. You can also, of course, make inferences about the impact of drug-drug interactions. And, this has made its way in the literature, also in terms of CP guidances. And, people were very excited about means and mechanisms of characterizing this 2 to 4-fold variability that we see in PK. However, what does this mean in the PK/PD context? Because if you look at the respective susceptibility of the infecting organism, what you see is that you can have like an up to 80 to 100-fold variability in the ability to respond to the drug. And so, in theory, you can, of course, make out scenarios. And we did this here in a publication in 2018, where we computed the probability of target attainment, and that is achieving a trough concentration over MIC ratio greater than 2. And based on that, of course, you can plot out various scenarios where you say, okay, so what happens if I have a susceptible organism? And so in this particular case, the probability of target attainment is close to 100%, irrespective of any drug-drug interactions or any genotype that you may have. On the other hand, of course, if you're dealing with a highly resistant organism, then, again, it doesn't matter that much either, because concentrations are too low in any case to be effective. And you're really talking about a sweet spot in the middle here, where variability in pharmacokinetics matter to help then classify between subgroups of the population who respond differently to treatment. And so, why would I say that? Well, because if you look at clinical practice, when you ask what's the MIC or what's the susceptibility of the organism, the answer is usually, I don't know. It takes a long time to culture things out. And even if I culture them out, I recognize that there is a certain measurement error just by the means of MIC determination as a twofold error. And so then, of course, that begs the question, are we closer to 80% or are we closer to 40% in terms of probability of target achievement? And so again, you need to put all of these things together when ultimately making a decision. So what are we willing to assume? Again, drawing an analogy between PBPK models and in this case pharmacometrics or population models. And what is really the impact of the research question on model complexity? So in other words, how complex do we really need to go to answer certain question? I mean, of course, you can characterize all organs, blood flow, or the whole body in terms of organs and blood flow, if you wish. But people, if you think about it, are perfectly comfortable, and I borrowed a slide from Jeff Barrett, by also lumping these things together in a population or analysis approach using compartmental models. And ultimately, if you use PBPK, we focus on process characterization versus if you look at POP-PK models, you really worry about parameter identification and estimation and identifiability. So, for example, to then stratify for subgroups of the population. So, what is the impact of the knowledge base? So again, to take this thought a little further, there is the common perception, of course, that PBPK models, are somewhat simpler, also structurally simpler than QSP models. I disagree with that. I mean, if you wanted to write out all the differential equations and all the parameters that are underlying a PBPK model, I think we would at least be at par with any QSP model out there in the literature. However, the difference here comes in, at least as far as I'm concerned, that you have you're trying to characterize with a PBPK model ADME processes that are typically very well known. On the other hand, if you have QSP models, also as we heard this morning, the underlying pathways and pathophysiologies may not be as well known, and so we're getting then into the realm of structural model complexity, model identifiability, and so on and so forth. At the same time, recognize that PK in isolation doesn't mean a thing, PD in isolation doesn't mean a thing, so we need to connect it again to PK-PD relationships. For PBPK models, oftentimes you feed then the information into relatively simple pharmacodynamic models, so log linear Emax or sigmoidal Emax models, and for QSP models, and this is just an example here. Oftentimes you use a relatively simple front end, so for example, 1 or 2 compartment models to characterize the impact of drug exposure. So the key question is here really, how confident are we in our modeling assumptions? And we heard before lunch that obviously, generating a lot of data, particularly in the rare disease space for characterizing natural history, is important, and I would agree with that. So important here, the next point I'd like to make is that obviously all of these models again are intended to integrate what we know, and I picked a relatively simple example here to see how we can use this information. And I picked Plavix or clopidogrel as an example because it had also received a black box warning from FDA, and it's a widely used drug throughout this thing. So it had to receive a black box warning for Sub2C19, poor metabolizers, potentially not receiving a full treatment benefit. And so let's see, where we are in terms of the overall importance of this polymorphism as determined by global sensitivity analysis. So the question that we had, can we put like everything that we know together and then use that to identify clinically relevant sources of gene subject variability? And so what we did here is developed a relatively simple physiologically based pharmacokinetic model by scaling up in vitro enzyme kinetic levels, connecting this through blood flow for both the parent as well as intermediate and active metabolites that then ultimately get linked to platelet reactivity, in other words, pharmacodynamic response. And so then we ran global sensitivity analysis, and you can obviously do this for the parent, the active metabolite, but then ultimately also the pharmacodynamic response. And what you see here is that the Sub2C19 intrinsic clearance that is subject to genetic polymorphisms came out as number 4 in terms of the list of importance. Number 3 is the fraction absorbed, not really surprising because it's a BCS class 2 compound that has dose-dependent solubility, but then also the baseline platelet reactivity, highlighting again that it may make a difference if you give the drug to a 20-year-old or an 80-year-old. And the most important driver was here carboxyl esterases 1, because they're the main bioconversion mechanism. And there is emerging evidence in the literature that there are genetic polymorphisms available in CS1, which reduce this pathway by 20 to 30% in terms of the ability to convert drug into the active moiety. The other thing that was interesting to see here is if you look at the x-axis, so the total sensitivity index, it basically gives you a percentage of variability that you explain. And if you compare this percentage here, so around 5 to 10%, this is coinciding with what's reported in the literature to the relative contribution of variability to 2C19 to the overall ability to respond. So that was actually comforting to see that we can translate mechanistic knowledge into outcome here. The next fact I'd like to highlight is, again, for more complexity and what do we need to know. And I picked a relatively simple example here for such a glucose and insulin model. And so the point I'd like to make here is that, of course, you can set up a model and, rightfully so, to characterize changes in glucose and insulin over the course of 24 hours, and you can characterize this data beautifully. However, again, coming back to the research question, is that really what we always need to focus on? And so why would I say that? Well, because if you look at what's then happening, and this is coming out of a collaboration with Eli Lilly, what is happening in terms of long-term clinical outcome, and I'd like to focus your attention here on the right-hand panel, the middle graph. You see, of course, HbA1c as the approvable endpoint that, and I give an example here for treatment with thiazolidinone, so an insulin sensitizer, that you can distinguish between different parts of the exposure response curve. And that is that the first part here is, of course, then governed by the extent of your treatment effect and also maximum treatment effect. After 3 months, you read out your HbA1c as the pivotal endpoint. And ultimately, at least if you're on treatment with this insulin sensitizer, you have basically establish a new homeostasis at a steady state following drug treatment. However, if you switch treatment, for example, to sulfonylurea, so an insulin secretor, you see that all of a sudden, the treatment response changes in a sense that it starts to wear off over time. And what you can see here really that this is the result of disease progression moving forward. So in other words, as we're losing beta cell function, and again, employing knowledge about pharmacology, that's not really surprising because, essentially what you're telling your pancreas that is already under stress conditions, okay, I know you're stressed, but why don't you work a little harder? And, at some point, the beta cells are going to give out and lose beta cell function. And it was also interesting to see that if you look at the diabetes community, that there are thoughts now to not only use insulin as a last resort treatment, but also treat early with insulins to conserve beta cell function and then delay progression of disease. And again, this can be also supported through mechanistic insight and modeling to look at these different treatment effects and treatment options. Obviously, you can expand this further, as we have done, to also account for the effect of diet, exercise, and surgery on body weight and insulin sensitivity. All right, so let's switch focus on what I think we should do next. Number one, again, I think we should focus on building consensus rather than silos. So it's not pharmacometrics versus systems pharmacology. It's not PBPK versus QSP. Again, in my mind, it's a modeling continuum, not a modeling competition. We already talked a little bit about the potential for model reusability this morning. I think the fact that we were able to reuse models, in the PBPK realm, for example, for evaluating drug-drug interactions was tremendously helpful. And I think we should probably try to go in the same direction for QSP models, so to establish disease modeling platforms in the context of use. And I'm perfectly cognizant, however, that this may require the use of multiple drugs, potentially competitor drugs. So I think this is more of a broader effort than any single institution can, for example, handle. And ultimately, I think we should foster transdisciplinary approaches, as highlighted earlier again by Dr. Sine and outlined first time in PDUFA 6. For example, through comparing notes between model-informed drug development approaches and real-world evidence, and we published on that before as well. And I just want to leave you here with a small case example to finish up my talk, to show that this can be done. So the example that I picked, and there's a variety of examples that I could have used, is dabigatran. Dabigatran is a direct-acting oral anticoagulant. This PGP substrate has a low oral bioavailability and is primarily cleared via the kidneys. And dose reduction is only recommended in subjects with renal impairment. So, however, when we looked into the literature, then we see that DOACs are oftentimes co-administered with, for example, PGP inhibitors, for example, in the area of HIV. And what we found here is then when you compared dabigatran versus rivaroxaban versus apixaban, that you saw a substantially increased bleeding risk in subjects with normal renal functions inhibiting PGP inhibitors. So a dose reduction may actually have to be considered. And so ultimately, the question that we asked, okay, that's fine, but is PGP inhibitor equals PGP inhibitor? And what about the degree of renal impairment? And so we did some further analysis using exposure response and PBPK modeling to simulate this out. And ultimately, what we found is that dabigatran should be avoided in subjects with severe renal impairment when co-administered with ritonavir or copicistat, such as in HIV, which is oftentimes given to HIV patients. However, PGP inhibitors is not equal to PGP inhibitors. So subjects with moderate renal impairment receiving ritonavir, either we should separate the doses between ritonavir and dabigatran, or reduce the dabigatran dose to 110 milligrams twice daily. For copicistat, those should be further reduced to 75 milligrams twice daily, and subject with moderate renal impairment. No dabigatran dose adjustment is needed in subjects with normal renal function receiving ritonavir, but it should be reduced to 110 milligrams twice daily in subjects with normal renal functions receiving copicistat. And with that, I thank you for your attention.
Thank you, Dr. Stephan Schmidt, for a very exciting presentation. In the interest of time, I would like to turn it over to Dr. Brian Smith.
All right, thank you. So I'm going to talk about development and application of a QSP model that was used to support extension of dosing intervals for nivolumab across indications. And I did want to give a little bit more background before I jump into the case study, right? So I want to give a background on mechanistic modeling as applied at Bristol Myers Squibb, the background on the model-informed drug development strategy that was used to enable new dosing regimens for nivolumab. And then I can talk about tumor receptor occupancy modeling for nivolumab, then development of the approach, mechanistic insights and key results, considerations for reporting model results, and then conclusions. So there are a number of challenges that mechanistic modeling can help with. It can help with target verification, with understanding optimal drug properties and molecular design. It can help with translational strategy. It can help with dose range for first in human. It can help with dose range for phase 2 and establishing proof of concept. There are also applications for confirmatory and understanding for phase 3, and then also post-market applications, which are the focus of the case study I'll go into in a little bit. And so another point to make is that mechanistic modeling approaches are fit for purpose. So we can start from from pathway models, mechanistic PK models, receptor occupancy models, mechanistic PK/PD models, and platforms and disease models. And so the modeling approach that we optimally take would consider things like timelines, what the scientific questions are, and the available information and data, right? And maybe one other thing to point out here is that larger platform models, Stephan was drawing the parallels with PBPK models actually being quite substantial under the hood. So I think for QSP disease models, it doesn't mean that they can't be fit for purpose. We can look at the actual pathways, update them as needed for a particular scientific question, and also, just as importantly, think about the calibration, validation data we have, and what the strategy is for establishing confidence in particular predictions from that model. So I wanted to give a little bit more context here because, right, the receptor occupancy modeling work was one component of multiple strategies that were used to support the new dosing regimens for nivolumab, 240 milligram Q2W, and especially 480 milligram Q4W. And so motivation for this work was, to prepare to have a flat dosing option for simpler dose preparation, and then also the Q4W extending dosing frequency provides a convenient treatment option for patients and healthcare professionals. So many clinical data were available for other regimens, and model-informed drug development was critical for establishing the safety benefit for 40 milligram Q4W. So a number of approaches were applied. There was a comparison of exposures via modeling. There was a well-established population pharmacokinetic model. Comparisons of key PK outputs were used. There's a comparison of key regimens. There was also safety bridging evaluation with modeling. There's an assessment of pharmacokinetic exposures for 40 milligram Q4W and benchmarking against some of the data that was available for higher Q2W doses, so 10 milligrams per kilogram. Then exposure response analyses was also done to predict the probability of safety events. And then efficacy bridging was also done via modeling. So exposure response analysis was done, especially for tumors where we had rich data. And then RO modeling was used as an additional component to support those analyses and to better enable extrapolation across more tumor types. And we used different scenarios, base case, best case, worst case, to encapsulate some of the different biophysical factors that were anticipated to drive target engagement across different tumor types. And so a substantial portion of this work was published back in 2019, although I think that the RO modeling wasn't described at quite the same level of detail that I'm going to go into today. So, and additional background on the case study. Right, so modeling approaches were applied to supplement gaps in clinical data. So these are an assessment of some of the data that was available, and this is available on the FDA website. FDA also published a paper on this analysis. And right, if we look across tumor types, we see that many tumor types have a flat dose response, and we do see dose response in some tumor types. Exposure response modeling was also done, and similarly with the exposure response modeling, right, it looks like there's a flat exposure response relationship for melanoma and renal cell carcinoma, but there was a trend towards higher ORR with increased nivolumab exposure in non-small cell lung cancer, especially squamous non-small cell lung cancer. So I think there were differences were suspected in some tumor types between 1 milligram per kilogram Q2W and 3 milligrams per kilogram Q2W dosing. Predicted ORR was similar within the exposure range of 3 milligrams per kilogram Q2W and dosing of 40 milligram Q4W. So that's promising, but there are additional pieces where mechanistic modeling can help to address gaps, right? So here, for example, additional confidence in the exposures, especially early exposure for 480 milligram Q4W are acceptable. And so, another important point to make is that right, despite these differential responses we see across tumor types, peripheral RO is generally saturated. And I think there's a lot of controversy in the literature rooted in analytical methods about whether peripheral receptor occupancy really is saturated for nivolumab. So this is a nice data set, and this is available right publicly. You have to dig a little bit for it. I got this one from the EMA website. So right, in this study, there was a fresh whole blood assay where there was about 100% RO. So peripheral RO was largely saturated even at low doses, 0.3 milligram per kilogram Q3W. So saturated peripheral RO at low doses, and I'll maybe add that, right, this isn't just renal cell carcinoma. This is where we have the nice published data set, but I think this is broadly applicable, kind of independent of indication that the peripheral RO is saturated at very low doses. And that coupled with these differential responses that we're seeing at 1 milligram per kilogram Q2W, right, that suggested that there is heterogeneity in tumor types, with non-small cell lung cancer representing a tumor requiring higher exposure. And also, maybe this is a little bit obvious, but I think it maybe bears, just repeating, the blood is not necessarily a good tissue to establish proof of mechanism and engagement at the site. So mechanistic bonding could also provide a rationale for why the site of action may deviate from the blood and prediction of tissue target engagement. Okay, so this is the RO workflow. So basically, there were a number of data sets that were employed. Some of them were new. We did go back and run new in vitro assays to establish some additional molecular and cellular parameters. There was also pharmacokinetic data sets and literature research, which you could consider prior, although we did have to do a lot of work to to find those papers and find relevant parameters and read them and investigate the data that was available. And then we come to base models. So there were mini models for nivolumab parameter estimation that were new. There was a POP-PK model, which was I think pretty well established, but we used that as one input into the QSP model. And then tumor transport, PD-1, PD-L1 ligand engagement was basically integrated from information in the literature. And then we go to integration checks and sensitivities for tumor RO, right? So we have a combined integrative model. We check the peripheral receptor occupancy predictions with the data, and then we do multivariable sensitivity analysis for tumor RO on day 28 following an initial 40 milligram dose. And then from there, we develop a number of scenarios, base case, best case, worst case, and then we can assess what the predictions are for those different tumor scenarios. So going to the first point, right, the new in vitro data sets were generated for estimating antibody avidity. This is a cartoon of the simple model that was used. And so this was combined with SPR data of antibody Fab fragments to get monovalent kinetics, right? So the way the assay was configured using the Fab fragments, we had high confidence. We actually, I think at one point we immobilized the PD-1 on the chip. We immobilized the Fab fragments on the chip. We got pretty reliable monovalent on and off rates for Ka and Kr kind of from this cartoon. Then a new cell binding assay was developed to assess affinity and avidity characteristics for antibodies, right? So I think the key point here is the avidity consideration, right? That you really do want to look at binding on a cell to assess avidity in a more physiologic context. So the assay uses directly labeled primary antibodies and quantitative calibration beads to establish a number of sites per cell or sites per square micrometer given kind of the cell surface area. And so modeling and fitting the assay is critical to derive expression and avidity parameters. So KXA is one parameter that came out of that cell-based estimation, right? Actually simulating the in vitro assay. And so that's antibody cross-linking rate, and the dimensions are area per number per time. It's a two-dimensional cross-linking rate, and then also RT, which is the expression parameter. So here we took it on an area basis, number of molecules per unit area, right? And these are what the antibody binding curves generally look like. And we have run some antibodies where we see the second peak appearing. It does really depend on the properties of the antibody. And we describe this in this publication in CPT PSP. So here, an internalization assay was also developed. So we used a fluorescence quench methodology. So it's nice because there's a surface signal to normalize against, and we ran it both in continuous mode, pulse chase mode. And this was similar where we simulated the in vitro assay to derive parameters to then translate into the QSP model and guide the sensitivity analysis. So this was similar to avidity measurements with fitting the simulations to the data following the experimental protocol in the simulation. And then the model accounts for antibodies essentially falling off in pulse chase mode. right? So that's this dotted line here in pulse chase, right? The signal decreases very rapidly, but through simulations, this is substantially attributed just to the antibody kind of dissociating when you wash and have a very low sort of concentration in the media that the antibody basically re-equilibrates with. So this is the RO model structure. So pulling all of these different pieces of information together, right, and implementing a cell binding model, right? So there is the two-compartment pharmacokinetic model, and this was a POP-PK model. Under the hood, we had time-varying clearance, used the covariate information, and used actual pharmacokinetic parameter and distributions to give real sort of variability in the projected systemic concentrations. Then there was a tumor transport model. This was based on previously published work. So I think there are a couple of different ways to represent a tumor. We went with strategies that have been published by Schmidt, Wittrup, and Thurber. And this tumor captures enhanced permeation and retention effects and the impact of elevated interstitial pressure. And then we have the cell binding model, right? So this is, we have cells in the blood and the tumor. This incorporates considerations like affinity, avidity, membrane transport, and then synapses with 2D confinement and ligand competition. And there are additional good references for the pathways. Down here, Jennifer Linderman has published related work, and also Michael Dustin has some very nice work on differentiating 2D and 3D reaction kinetics. And there are other papers that reference some of the PD-1 and ligand interactions. So model check. So this is our first model check, and that's essentially that we can capture the saturation effect of receptor occupancy in the periphery. So here, across dose levels, our assay RO was essentially saturated. Simulated RO was essentially saturated. Some very sort of small differences in target engagement across these dose levels. The IQR for the assay was larger than the IQR for the simulation results, but I think that's due to some intrinsic assay specific features that we're not capturing in the model. Okay, and then I ran sensitivity analysis. And so this is a list of 16 parameters that were run. And basically these were selected because there are areas we knew that we were going to introduce variability into the model. Maybe there was a little bit of uncertainty in the literature, and also they're looking along different pathways. So these are different processes like internalization, pharmacokinetic properties, transport into the tumor, receptor ligand interactions. So these were kind of selected to span the biology. And so running the sensitivity analysis, this was done by modeling intratumoral RO at day 28 following a 40 milligram dose of nivolumab. We did check convergence of the Sobol indices. The open circles are the median from bootstrapping, and the error bars indicate the 95% confidence interval. The intratumoral RO in this case was most sensitive to fraction of PD-1 expressing cells involved in contact with ligand expressing cells, ligand expression, ligand association, and the clearance rate. So this is one area where the physiology, I think for target engagement isn't necessarily consistent with different classes of molecules, right? It wasn't the internalization parameters that were showing up here as being extremely influential, kind of given some of the in vitro data we had available to constrain that pathway, right? So many physiological factors are expected to vary between tumors, but they weren't necessarily identified as being very influential on the intratumoral for nivolumab. So then we simulated projected target engagement for tumor RO for the new dosing regimen under a variety of scenarios. So base case, worst case, best case, 3 milligrams per kilogram Q2W as a reference for our gold standard, 240 milligram Q2W for the flat dosing, and then 40 milligram Q4W for the flat dosing and extended dosing interval. So here the population parameter, the pharmacokinetic parameter distributions were set according to the POP-PK model. Tumor parameters were varied for scenarios, so you have different cases if their sensitivity index exceeded 0.05 from the previous slide. And so these different scenarios are representative tumors with different biophysical characteristics. And then the worst case scenarios were needed to drive lower tumor RO. And so here are the same, here we're looking at box plots of the data, and we're looking at day 14, day 28, and the time averaged receptor occupancy over day 0 through day 28, base case, worst case, best case. And then the actually looking down to 1 milligram per kilogram Q2W, which wasn't in the previous time domain analysis, 3 milligram per kilogram, 240 milligram Q2W, and then 40 milligram Q4W. And so what we do see is with the worst case, we see differentiation between the best case and the worst case. So, right, looking between 3 milligram per kilogram and 1 milligram per kilogram, we have a minus 0.5% difference in median receptor occupancy versus minus 9.2% difference. So it looked like if we go to a worst case tumor, it is very possible we could be driving differences in efficacy at the lower doses where we had clinical data. So the most sensitive parameters to define the worst case are specific to tumor pathways, cell contact. So essentially the model is suggesting we should be maintaining high peripheral RO even with this lower target engagement in the tumor. So the 40 milligram Q4W regimen exhibits higher intratumoral RO at day 14 than 3 milligrams per kilogram Q2W, and marginally lower but comparably high intratumoral RO at day 28. And so the high time average intratumoral RO is maintained with 240 milligrams Q2W and 40 milligrams Q4W dosing relative to 3 milligrams per kilogram Q2W across the various scenarios representing a variety of tumor types, right? So the RO QSP model does support that 240 milligram Q2W and 40 milligram Q4W regimens should not compromise efficacy. And the time average RO will be maintained over the first 28 days for a variety of scenarios with different tumor characteristics. And so, just thinking about the kind of the mechanistic insights, from this model and kind of what we do with it, right? I think that this is interesting because, right, there's a physiological consideration here. If an antibody blocks a receptor ligand interaction in immune synapse, as with checkpoints, if you're in a similar quantitative regime, then dissociating the tissue would, essentially relieve the competition. There's another consideration. It's going to release drug from the blood, right? If you had a strong concentration differential between your tumor and the blood. Here, I don't think that impact is that dramatic, but it'll also make it easier for the antibody to bind the receptor because of relief of these competition effects. So this suggests that the RO at the site won't necessarily be well reflected with methods that dissociate tissue. So I know when we talk about measuring RO at the site, one method that's often proposed is flow cytometry, and it's suggesting that we might not want to use flow cytometry. We might want to think and basically take the tissue, dissociate the cells, and then shoot them through a flow cytometer. That's going to change. That could change our measurement quite dramatically. So, right, if we want to think about validation, the site of action, right, there are some different strategies that could be used. Imaging and IHC-based methods with intact tissue sections. I think that has the potential to address some of these methodological challenges. And more of a clinical challenge, if we have a post-marketed therapy and we really want to go back and validate, quantitatively certain outcomes from the model, and we want to capture the sensitive regions of the dose exposure response curve. And so if we're doing that, are we kind of denying patients the benefit of the higher sort of dose that they're going to need to really derive the benefit? Right, but I think there's some practical ways in designing the protocol that patients might be able to, that they should be able to ultimately get the dose that they need, but we could do, some initial work to kind of test these hypotheses. So data availability guide the application strategy and can guide the decision-making, right? So this case highlights differences in strategies for mechanistic model application. So what I showed was a scenario-based strategy where we built confidence in the model. I didn't have direct measures of key outputs, target engagement at the site. So then what was done was there was a development of base, best, and worst case scenarios, from the sensitivity analysis to extrapolate based on that mechanism. Another paradigm I think can be targeted prediction, right? But there we need validation data more directly related to the model outputs of interest. So it might be challenging in this case, getting that right target engagement data from the site. But if we wanted, a mechanistic model that was going to reproduce, ORRs, then that might, right, that'll have a different set of challenges. But getting the, ORR for related therapeutic intervention should be, potentially less of a challenge. So I would say here, just a, nice thing to consider is that data availability can be a bigger influence than model size for the strategy of how you actually go about deploying and assessing the reliability of the model for your application. So I think another quick point I can make is about report format and submission guidelines, right? So when this work was done, this was, back around 2017, 2018 timeframe. There wasn't, much guidance on QSP report formats. There was, input from PBPK, right? So at the time when the work was started, I think we just had the right the draft guidance, and that was used in kind of setting up the to help guide how these sections on the report were set up, and to some extent form follows function, or right with the reporting format, function follows form. Sections in your report are going to be a part of your analysis, or you're going to have to say why you chose not to do that particular analysis and justify it. Another point is flexibility in file formats are, I think, still pretty important for submitting QSP models. At least this is maybe just my opinion, but right, we don't have, I don't think we're at the point where all of our analyses fit into nice, simple, flat text files. And for this work, we did have a directory structure, organized files. We had a version control toolbox with folders. The model format was proprietary. It was binary. It was an SB proj file, symbiology model. We did make it really easy to run. If somebody wanted to reproduce all of the analyses, we went through and set up like a runall.m script that you could just type that, and then it would reproduce all of the figures that were and analyses that were in the report. And so this was submitted on a zip archive. So you can imagine directory structure, binary file formats. How do you transfer this to someone? So we did have the option of using a physical disk, and so that's what we took, because I think some of the ESUB submission formats aren't necessarily this flexible to be able to do things like with zip archives and binary file formats. And right, so today I think we're different than we were in 2017, 2018. So there's a bit more guidance in what could go into a QSP report. So right, it's still, we don't have a single sort of published standard from a regulatory agency, and this needs to be this section and this should be this section. These are analyses that go into each of those sections. But I think there are good guidelines to think about what the community is starting to expect, right? So we did have the FDA Industry Scientific Exchange where we touched on some of these items. You can also look at other case studies. So, the other speaker from Sanofi, Susana, Dr. Zaph, her team has an FDA sort of evaluation available online. You can go there and look at what reviewers were actually looking for to see what information you need to make prominent. And then there's also, there was, some information from CDRH. They did propose, they did have a draft proposal for assessing the credibility of computational modeling and simulation and medical device submissions. And so I think you can, maybe look at that and use it as a starting point. Although, for QSP and drug development, things could vary a little bit, but I think to get some general ideas that also can be helpful. And then again, moving forward, I hope that we can maintain some flexibility in file formats for submission. And so in conclusion, mechanistic modeling can be applied to address a variety of questions along the research and development pipeline. MIDD was critical for the approval of nivolumab 480 milligram Q4W across tumor types. Mechanistic modeling was employed to develop additional confidence in a new dosing regimen, enable a pan-tumor assessment and extrapolation across cancers by accounting for critical biophysical differences, encapsulating rare and more common tumors, and then explain potential differences between the periphery and tumor tissue. So just another brief point, which maybe is clear, but I can reiterate, mechanistic modeling is not limited to early research and translation, and mechanistic modeling has the potential to play a larger role in drug development, including post-market applications and BLAs. So with that, I would like to thank the organizing committee and session moderators, Dr. Bai, Dr. Shuk, Dr. Stinchcomb, and Dr. Earp, and also a number at BMS that have been engaged in this effort. Tunde Bello has been a continual supporter and was helpful in this work. Amit Roy has been a great senior colleague and provided helpful feedback. Former BMS employees, people in discovery that would sit down, run those protocols, those new experiments multiple times, and make sure that we're getting the data out of their experiments and fine tuning the protocols. Christine B, Minhua Han, Michelle Kuhn, and former colleagues Yugan Cheng, Tarek Leal, and also collaborators at the University of Maryland. And so any follow-up, you can send me an email, brian.schmidt@bms.com. And maybe just another point is our QSP and PBPK team is hiring. We have multiple roles. We have QSP roles, we have PBPK roles. We really need strong industry experienced associate director QSP modelers. We have some exciting new initiatives with the thematic research centers at BMS. And we're looking for strong individuals that might want to get experience prioritizing a portfolio and taking on a broader responsibility. And with that, I'll stop.
Thank you so much, Dr. Brian Smith, for very interesting presentations. We are very behind, and so what I would like to do is that if you have any questions, just text the speakers, and we will now turn it over to Dr. Susana Zaph. Thank you.
Thank you. Can you see my slides?
Yes.
Great.
Yes, I just need to be put in presentation mode.
Yes, it's coming. Can you see it?
Perfect, thank you.
Perfect. Well, thank you so much. I want to start by thanking all the organizers of this workshop for the invitation. It's been a quite delightful day hearing about rare development and QSP. My name is Susana Zaph. I lead the translational disease modeling group and that covers rare and neurotherapeutic indications at Sanofi. And today I will be presenting a QSP-based analysis that we actually included in the BLA for one of our drugs, lipidase alpha. We basically use this analysis to quantify how much similarity of disease and response was there between pediatric and adult patients in a very rare disease called acid-sphingomyelinase deficiency. And I also want to point out and give a shout out to my team, RJ, Mandy, and Chinchala, who are the machines behind this operation. So I'm going to start by giving you a very brief overview of what is acid-sphingomyelinase deficiency. It's a rare monogenic lysosomal storage disease. It results in an enzyme deficiency that basically prevents the breakdown of sphingomyelin into ceramide and leads to this toxic accumulation of sphingomyelin in tissues that are of reticuloendothelial origin, such as the lung, the liver, the spleen. And that accumulation drives organomegaly in spleen and liver, and also a significant loss of lung function. There are different types of presentations for acid-sphingomyelinase deficiency, depending on the degree of the enzymatic deficiency, with the most severe being the type A, and these patients tend to have a significant reduction in enzyme activity that leads to a significant accumulation of sphingomyelin in tissues, and not just in the visceral tissues, but also accumulation in the CNS and peripheral nerves. And these patients tend to have a very rapid progression and very early onset and high mortality. Sorry. Lipidase alpha is a recombinant form of acid-sphingomyelinase that it acts as an enzyme replacement therapy, and it has recently been approved as the first treatment for the non-CNS manifestations of acid sphingomyelinase in both pediatrics and adult patients. So just to give you a very brief overview of the clinical development, this is a very rare indication, and thus it contains 5 different clinical trials in adult and pediatric patients, with 1 adult trial that was a placebo-controlled pivotal trial, and due to the rarity of the disease, 1 pediatric trial with an open label design. Also of note is that due to the rapid progression of observed in the type A patients, these were not included in the open label pediatric trial. And so there was there's been reported that there was substantial evidence of effectiveness of response of olipudase in the adult pivotal trial. And the idea was to develop a modeling and simulation package that would complement the pediatric data from the open label trial. And that included traditional POP-PK-PD approaches and ER relationship analysis, but also included a QSP component that led to 2 objectives. One was to use a QSP approach to quantify how similar this disease and the therapeutic response was between the pediatric and the adult ASMD patients. And 2 was to predict the efficacy of olipudase sempozyme in patients that were not included in the clinical trial, the type A patients, which have the most extreme enzyme deficiency. So with that, I'm gonna just give you a very brief overview of what the model looks like. So the model is multi-scale and modular in fashion. It consists of a PBPK submodel that describes the PK and the biodistribution. And given how olipudase is actively uptaked into tissue via receptor-mediated mechanism, having this PBPK submodule was very useful in describing the tissue and lysosomal exposure. This was then connected to the molecular level description of the key enzymatic reactions that basically are needed to describe sphingomyelin accumulation, and also describe some of the biomarkers that were collected in the trials, such as plasma ceramide and lysosphingomyelin, which were either ceramide is a product of the sphingomyelin breakdown, and lysosphingomyelin is a derivative of sphingomyelin. So they're they're proxies for the activity, for the accumulation of sphingomyelin. And then we had also, the molecular level is also the molecular layer that we have the best or the most detailed description of the biology, given how that's the one that has the most available data to describe. Then the molecular scale is connected to all the plasma biomarkers, as I discussed. Then the sphingomyelin accumulation is the connecting entity that allows the description of the dysfunction that happens in the cellular layer, which it tends to be hepatocytes or splenic alveolar macrophages. And then this function in the cellular layer basically drives the organ level dysfunction, that in this case, in the model, we have representations of the spleen volume or the splenomegaly that characterizes the disease, and also the lung dysfunction as measured by use of DLCO measurements, which it stands for diffusive capacity of the lung for carbon monoxide. And this model was parameterized using a diverse data set, not just the data that is collected during the clinical trials, but also a number of data sets that are normally not used in modeling and simulation efforts. And that which makes this type of modeling very attractive and useful in rare indications, because you take into account all the prior knowledge that is known that is available on single myelin biology in this case. Also, a lot of knowledge that is available on macrophage function, and able to combine that with in vitro assays that describe the drug and animal models that also have some preclinical efficacy. So that allows you to really inform all the biological scales of the disease. And that then gets combined with the clinical data. When we were lucky that there was a natural history study that was available for us to use, and also the clinical trial data set. And at the time that this model was originally developed, only the phase 2 data set was available. But this type of effort really allows us to then combine all this information and all the formalism of the biology into what we call a cohesive, integrative view of acid-sphingomyelinase and olipidase response to create virtual patients. And just to zoom in some of the details that we include in this, I'm just going to zoom in into the molecular layer just to give you a peek. And as you can see, there's detailed representations of all the reactions that are affected in the disease, the conversion of sphingomyelin into ceramide, with the deficiency described by the residual enzyme activity parameter. So this is a parameter that varies from patient to patient because it depends on the type of mutation that these patients exhibit. There are certain mutations that result in a mild deficiency, other mutations that result in a very severe deficiency. So there's a natural variability in the degree of deficiency that these patients explore, and we were able to sort of capture that by including a parameter that describes how reliable the endogenous activity is acting in these patients. And also include the mechanism of action of olipidase, which is the supplementation of this activity that is derived by the olipidase. Also, other reactions that were included that were needed to describe the dynamics of lysosphingomyelin and ceramide. Another thing that I want to point out is that we talk a lot about how there's a lack of data in rare diseases, and that is a real headache, but we also have to be creative. And one of the things that we've done, not just for this model, but for other models, is that for these monogenic diseases where we have a clear understanding of the mechanism of disease, we can also leverage some of the healthy data, because a lot of these processes in healthy individuals that have normalized residual enzyme activity could be leveraged to inform some of these kinetics. So we do a lot of that, and I'll show you also a validation later on where we took advantage of that to validate some of the model. So since these QSP models are mechanistic in nature and not empirical, they contain processes and parameters that are biologically interpretable. And that kind of allows us to have a framework where we can basically benchmark the disease and response of adults versus pediatric patients, and really utilizing to the maximum the collected clinical data. And the way that we do this is to develop what we call virtual twins. So these are basically model representations for each individual patient enrolled in the olipidase clinical trial. They are calibrations of the model for each patient with their appropriate demographics, but making sure that their biomarker and endpoint profile is representing using exactly the same QSP structure, but varying a small subset of parameters that are basically become personalized parameter sets. And that allows us to account for some of the intrinsic variability of the disease presentation, and also helps us understand where the source of this variability comes. So we then calibrate the model to individual patient data in one cohort, such as the adult cohort, and we then are able to establish a expected degree of variability, how variable that biological process or parameter should be, and then compare that to the pediatric cohort. And the idea is that if we're able to capture the pediatric and the adult patient individual fits for PK, PD, and the clinical endpoints with comparable accuracy and with parameter values that are comparable, that would support this idea that there is mechanistic similarity between the adults and the pediatric patients, and allows us to really leverage these virtual twins. So how do we develop these virtual twins? So first we conducted a series of criteria that was based on a parameter sensitivity analysis, also biological knowledge, and also how reliable or available the reference values were from the literature. So parameters with high sensitivity that were known sources of biological variability, such as the endogenous residual enzyme activity, those were chosen as part of this individual calibration set that describes the virtual twins. The virtual twins were then performed in a sequential manner with cost functions and initial parameter ranges that were common for all patients independent of age. And also we try, when applicable, to make sure that those parameter ranges were confined to what we consider biologically meaningful or relevant ranges that were consistent with reported measurements. The calibrations also occurred in a sequential manner, taking advantage of this modular setup that we have for the model, so that the PBPK model was calibrated first, and the PK parameter for that was fixed, and then the molecular submodel was calibrated next against the plasma biomarkers, and then the organ level parameter was calibrated last. And we found that this type of sequential submodel calibration really helps with identifiability, and also allows the more empirical representations, so the more organ level representations that they now have as much mechanistic detail to be grounded by the mechanistic features of the molecular layer. So for example, the REA really determines then the type of parameters that can be calibrated to describe the spleen volume and the Delco dynamics for that patient. And that basically gives rise to the virtual twin. So we have this collection of virtual twins for both the pediatric and the adult cohort. And then, even though there has been a lot of discussion about how QSP models are amenable in principle to be used for this type of application, there hasn't been a lot of discussion about details about how that could happen. So we came up with some criteria that we felt would allow us to claim similarity. So, and the criteria was that the same model structure had to be able to describe equally well both the pediatric and the adult cohorts of patients with similar accuracy, meaning that the virtual twins, PK biomarker, and clinical endpoints are comparable in how well they represent the observed data. And with similar accuracy, as I mentioned. So if our model representation is poor in the adults but good in the pediatric side, that would be problematic. So we would prefer that the model accuracy was consistent independent of age. Then the other was that these personalized parameters that define the virtual twins are comparable across age cohorts, so that since they are describing biologically interpretable process, that they should, in theory, share the same magnitude at least and be comparable in distributions. And finally, that the parameter sensitivities for these individual patient calibrations should actually also be similar. So the influence of each parameter within that calibration should be similar in trend, as independent of whether it's an adult or a pediatric patient. And using these, but before we dove into the actual application of the model, we had to validate the model. And we did a significant amount of validations that required a lot of creativity, because as I mentioned earlier, there's a scarcity of data that it makes developing these models in rare diseases quite challenging. But the model was validated by comparing to external data that was not used during model development. And it allowed us to gain a lot more confidence on the model performance for each biological scale. So we try to create validation exercises that really tested each of the scales that we had represented in the model. And we performed several, but I'm only going to show you 2 approaches that we use, because I think really they talk about this use of creative data to really validate your model. The first one was, since this is a monogenic disease with a clear cause is an enzyme deficiency, we could leverage data sets from healthy individuals if we simulate a population without enzyme deficiency. So if we move that REA parameter from whatever 30% of normal to 100% of normal, and that's basically what we did, is we develop a virtual population of healthy subjects constructed based on the adult virtual twins, where the enzymatic deficiency was normalized, and just as you would assume healthy individuals would be, and then predicted the untreated plasma ceramide or plasma lysosphingomyelin, and compared it against reported data sets from healthy individuals. And as you see, we saw a good agreement between the 2. The next thing we wanted to validate was a lot of the pediatric representations that we had. And as you can imagine, that there was even less data there. And we there's very limited number of pediatric patients that we had access to. So splitting the pediatric cohort into a training and validation data set was just not feasible. So instead, we generated a virtual population using a bootstrapping approach. We basically, what we did was remove the parameter set defining the virtual twin of the patient of interest from the parameter matrix, and used to generate the respective virtual population with the rest. And this ensured that the virtual population that was created was not being informed by that specific patient. And then we simulated the resulting virtual population, and we selected the top 10 most similar patients at baseline of their biomarker profile, and assigned those as the virtual population for that individual patient. And what I'm showing you down here is the predicted versus observed plots. As you can see, that created a fairly good match between the observed data and the predicted data, really increasing our confidence on the model. But going back to this criteria that we had, where we had the 4 quadrants that we wanted to satisfy, the first quadrant, as you recall, is this idea that the same model structure should be able to capture equally well pediatric patients and adult patients. And we did this by looking at how the fits, which are the lines, versus the observed data, which are the dots, for different patients look. And here is just a subset of the available patient data set, but we have all the way from infants to adults. And as you can see, based on the magnitude of the baseline values, there are also very diverse severities. And what you can see from here is that when we compare the virtual twins to their respective observed data, the same model structure could capture the biomarker and an endpoint profile regardless of age or severity. The next thing we did was to actually quantify how much of an agreement there was between the virtual twins and the observed data set. And we use goodness of fit plots. And as you can see here, when we have the overlay of the pediatric data sets versus the adult data sets for the observed versus predicted, you can see that there's a huge amount of overlay. And also the bias is pretty similar. And we use a number of accuracy metrics to quantify this. And they seem to be comparable across cohorts. We did, I think, Pearson correlation coefficients. We did also normalize root mean square error, and these were similar in both pediatric and adult patients. The next thing we did was to analyze the actual parameter sets from each individual virtual twin. And as I mentioned earlier, there were 12 parameters that were used to define the virtual twins. And when most of these were compared, the majority of them were considered similar based on a series of statistical criteria that we use to quantify this. We use the 2-sample KS test to compare the distributions, and we also did a comparison of the group wise median, sample median. And if you look at the distribution plots of the parameter, the individual parameter values for the pediatric and adults, you see that there's a huge amount of overlap that is agreeing with what the statistical analysis suggested. So, and this supports mechanistic similarity. The ones that were not similar, there were 3 of them. And we decided to test how meaningful these similarities were in terms of their response, in terms of their effect on biomarker and endpoints. And what we did is basically simulate a patient that had either the mean pediatric parameter value or the adult pediatric parameter value, and see what that gave rise to in terms of their biomarker profile or their clinical endpoint profile. And as you can see from these graphs, this is just an example. There were some minor changes in the plasma ceramide, but that did not translate into any other model output. The one that did have a significant effect was the residual enzyme activity. So when you look at the distribution of this parameter according to age, you see that the pediatric patients tend to have much lower values than the adult patients. So just to refresh your memory, the REA is the residual enzyme activity, and it represents the severity of the enzyme deficiency. So the lower the value, the more severe the presentation. And as you can see, these were deemed to be statistically different. And of note is that when we plot these individual predicted REAs for each virtual twin, you can start to see a separation when you plot them against the observed corresponding lysosphingomyelin, where the patients that have the lowest REA tend to have the highest lysosphingomyelin, which is not surprising because we calibrate the REA versus this data set. But what it is reassuring is that even though we don't calibrate the REA versus the spleen volume, you also see a very similar distribution where the most the patients with the largest spleen sizes tend to have also the lowest REA and tend to be also pediatric patients. So using this individual calibrations from the virtual twins, we derive a virtual population by sampling the parameter distribution from these subset of parameters. And what we found is, and then we plot them as the spaghetti plot shows here, color-coded, so that the pediatric patients in blue and the adult patients in red. And as you can see from these simulated spleen trajectories from birth to 60 years of age, it shows that basically there is a continuum of disease presentation. It's not well defined by age. There's a significant overlap, and it just seems like there's a continuum. We then went ahead and plotted in the same graph, observe treatment naive spleen size values along with their age of the patient. And then you start to see that there is these sort of quadrants where there are gaps in the data, observed data. So the young age patients tend not to have reported spleen volume, and the most severe older patients tend not to be very populated up here. And that it matches well what has been reported in terms of differences that have been interpreted by survivor and diagnosis differences. So the more mild patient tends not to get diagnosed until later in life, and the more severe patient tends to have lower survivorship. So this really highlights the power of having this type of approach. Finally, the last thing we did to satisfy the 4 criteria that I mentioned earlier was the sensitivity analysis. So for each patient, we did a sensitivity analysis for a baseline, for all the model outputs at baseline and also at week 52. And even though there is slightly bit different color in the sensitivity index for each of these patients, you can clearly see that the directionality of these sensitivities remains the same, independent of age. Also supported that the influence of these parameters on each patient seems to be consistent across patients independent of age. So I'm going to conclude today by just summarizing this analysis. This was, I hope that you're convinced that this analysis showed that the same pathophysiology that we included in this model with the similar parameter data sets can capture both the pediatric and the adult data sets in a similar manner. And we believe that this is supportive of mechanistic similarity of disease and response. And the key here is that we think that there's no distinct subpopulations that are defined by age, but it's actually a continuum of disease burden. And part of that is the variability of the disease severity and also the rarity of the disease that creates and I'm going to end also with what, as I mentioned earlier, this was submitted in the BLA, and after an intense review process, we received feedback that the reviewers of the FDA also agreed that these results support the approval of olipudase pediatric patients, since they do, in combination with the observed data, support this idea that there's mechanistic similarity in disease and response. But QSP is a it's a collaboration, as you can see from the slide. It is not possible to be done in isolation. And we were lucky that we have the support of the project team. We have the support of the digital team. We have the support of other modeling and simulation teams. And we've been incredibly lucky also by being able to approach key opinion leaders that have guided a lot of the information that we presented here. So I want to thank especially RJ, Mendy, and Chanchala, who have been relentless in working on this. And what I just showed you was the first time that Sanofi has included a BLA, a QSP in a BLA. So, and we hope it won't be the last. So I'll stop here and take any questions that you may have.
Thank you, Dr. Zaph. There's a question in the chat box. Please respond to the audience directly, and we will now turn it over to Dr. Earp for the panel discussion. Thank you.
Thanks, Jane. Working on getting video set up here. Okay, I'd like to invite all the panelists to join, turn on your video. And I'm gonna go through and introduce the panelists real quick. While I'm doing that, if you're in the general audience and you have any questions for this panel for session 2, which is overview of QSP and successful uses of QSP in drug development, feel free to submit any questions to the chat box. So for the panelists today, in addition to the 3 great speakers that we just had, Dr. Stephan Schmidt, Dr. Brian Schmidt, and Dr. Susana Zaph, we also have Drs. Raj Madabushi, who's the Associate Director for Guidance and Scientific Policy here in the Office of Clinical Pharmacology, Dr. Christina Friedrich, Chief Engineer with Rosa and Company, and Dr. Jane Bai, Expert Regulatory Scientist in Systems Pharmacology here at the FDA. So with that, I'm going to jump into some questions here, specifically in terms of contrasting PBPK and QSP and what we can learn from this. Often in science, we're able to learn about complex systems by building on the science of smaller components in that system. And the question for the panel is compare and contrast how PBPK could or could not be a small component of QSP and what we can take away from PBK for QSP. So certainly there will be gaps that cannot be addressed. And what are those? And how can we tie this back to verification and validation? What other modeling approaches for small systems could be used for gaining insights into establishing credibility of these models? So I know there are a number of components. I can refresh on that as we go through. So should I pose this? I don't know, Stephan, do you want to take a start at building on that? You're muted right now.
Justin, was it for anyone to start up, or you wanted to hear from specifically from someone?
Well, heard silence, so I called out Stephan, but Raj, if you want to take a crack, go for it. Anybody, this question is open to anybody.
That's fine. We can hear from Dr. Schmidt.
Which Dr. Schmidt?
Yeah, I think he was referring to me. So I tried to draw some parallels already during the during my talk, briefly highlighting parallels because as I said, I think there's learning curves in terms of what is acceptable, how is the field move forward, over the years, what are how do we get like buy-in from other stakeholders, maybe outside of the modeling domain, what are the lessons learned here? And I think there's a lot of parallels that we can draw between PBPK and QSP models. I think the original fear for PBPK models was they are too complex, and nobody's gonna understand that. But that turned actually out to be not to be true, because I think as long as we speak biology and physiology and pharmacology, I think the buy-in was actually quite easy for these applications. And obviously the value proposition was there relatively early on with respect to the potential of being able to waive clinical trials. And I think I've seen a number of applications today, in the last 2 talks, fantastic talks for virtual twins, and how this could be applied to rare diseases, for example. So I think that's the avenue we probably should go.
Yeah, so as part of that, do you see that confidence in these smaller or sometimes smaller systems need to be established before we can move to QSP, or simply it's coming down to the nature of the decision and how the model is being used, and that validation and verification can be done independently?
That's a loaded question. So I think I'd like to have also like Raj chime into this one here. I think there's no black or white answer to that. I think there's components both in terms of model complexity as well as the question answered to that. So I don't have a straightforward answer for that.
Yeah, if I can jump in on that particular question. We have always, in general, preferred parsimony when it comes to developing models, applying models for right reasons, because there are less amount of assumptions that would need to be taken into account. But as we accrue knowledge, it wouldn't make sense to actually ignore their presence for parsimony. So there has to be a fair bit of balance, and I'm pretty sure there are learnings from PBPK that can be drawn as parallels that may be projected into the QSP space. Hopefully, those kinds of learnings will translate at a faster clip than what it took for, let's say, PBPK. But one big advantage, I would say, with the PBPK evolution was the availability of information was never a challenge. There were so many PK studies that were there already to be able to leverage, fine-tune them, and also the ability of PBPK models to broadly be applicable, right? CYP3A inhibition could be applicable across many different kinds of inhibitors, for example, to be able to refine the understanding of the biology in the background. So that's where there is a small distinction, because there are going to be some shared aspects of biology in the QSP realm, but there are going to be some very unique aspects, especially if you're going to talk about rare diseases, where they are going to be uniquely applicable for a given setting. So I think it's over a period of time, as knowledge accrues, these are going to become more and more powerful in their applications. But Dr. Stephan Schmidt is absolutely right that that I think if there is a balance between the type of question that is being addressed and what are the gaps that need to be addressed, and how a particular application of QSP can play a role in de-risking or increasing the confidence in a particular exercise, probably there is a role for it to grow up in that space. Not all applications are going to be your holy grail applications where you're going to eliminate the need for standalone clinical trials. I think there are many other applications we should not lose sight of where they can be very informative to the entire drug development enterprise.
Yeah, maybe I'd like to add to this what Dr. Madabushi just said. I think, one good example for PBPK, and I think that's the main regulatory application at this point, is DDIs and maybe the general acceptance of paradigm compounds that could be used for evaluating the impact of DDIs, either strong or moderate index perpetrators, and accepting this then throughout the development context. I think that has been an important step forward, and I do not see any reason of why we could not do the same, also for QSP, particularly if we, as I said, if we have well-known pharmacology, pathophysiology, for example, monogenic diseases, and I think we heard a good example there, why this could not be used as a platform model. I mean, we have seen similar developments also for disease progression models, and I think CPATH has played a critical role there, and I think there's a certain connotation of regulatory acceptance of this type of models, and I don't see why we couldn't do the same for QSP here. So in other words, we don't need to jump that hurdle again, because there's precedence, and so maybe a bit easy to get to that point.
Yeah, thank you. We're a little bit limited on time given some of the run over that we've had, so I'm going to move on to a couple other questions that have been directed to us. One is, can the FDA comment on the size complexity of the QSP models that FDA has accepted in supporting an application? I can briefly comment, and I can send this over to both Susana and Jane and Raj if needed, but I certainly don't see any restrictions based on size, and in my understanding, this comes down to the nature of the application and how it's being used in what context. The model that Susana presented for lipidase alpha was applied for comparing parameters between pediatrics and adults, and I'll let Jane or Raj comment on any of the others. I don't know.
May I add that I think it is not related to the size or complexity, it's related to the credibility of the model, how in a summation, how a model structure and the data is used to calibrate the model and validate the model. And based on the context, questions of interest, context of use, and decision consequence, model inference, that the totality, the credibility that has been shown in the model. And that was the most important. Thank you.
Yeah, I do not have anything more to add on top of that. It's all dependent on the specific context of use and the question at interest. A complex model could be very useful for in the translational space because you have nothing but that as your best available information to go forward. A simpler model could be useful at a later stage. You never know where it plays out. It all depends upon those specific aspects, which were so nicely described in the morning session also in Dr. Zine's presentation. I think at the end of the day, it comes down to that appreciation of there is a time and place for application of various types of modeling activities to address different questions at hand. I think that was also highlighted in Dr. Stephan Schmidt's presentation.
Great, thanks. For the whole group and maybe in particular for Dr. Brian Schmidt, we have a question here. The use of virtual patient populations is increasingly common for quantifying uncertainty and variability in QSP modeling and simulation. And what are some of the successes and challenges and opportunities?
Thanks. And right, I think, rather than model size per se, I think, right, the main challenges are more related to, calibrating the, observed data well. And, right, and so model stiffness and timescales, I think, are a bigger sort of concern in, calibrating systems models than, actual number of equations, especially when trying to model phenomena across timescales. It's very dependent on, the types of calibration validation information that you have. And right, I think there are a number of approaches that can be applied to address these sorts of challenges, right? The most obvious being more compute power. Another one is, improving simulation times by improving simulation timescales, using things like quasi-steady state approximations where they make sense, other simplifications that you can improve the model performance. And also, more efficient calibration algorithms, better numerical solvers and approaches like machine learning and surrogate models, right? So all these challenges are tied to opportunities. And I think there's a lot of substrate for improvements to some of the calibration strategies, leveraging not just, say, for example, Bayesian methods that are embraced by the systems biology community, but also, say, frequentist statistical methods that can be inspired by some of the improvements that have come from algorithms that have traditionally coincided with Bayesian statistics, but can also be used in a frequentist context.
If I can maybe add a comment there, particularly thinking about the context of rare diseases, I think Dr. Zaph's presentation had a very nice example of this, where I think the opportunity with the virtual patients and virtual populations is also a very careful and thorough examination of your hypotheses, and especially in rare diseases where you have limited data, really digging into the science and trying to understand what are all the different ways that your model could actually fit these limited data is extremely valuable, because you may find out that some of the hypotheses that you had going in actually turn out to be not consistent with the evidence, even though you may have scant evidence. Once you put it all on a dynamic timescale and quantify everything and compare apples to apples, you may be able to eliminate some of the hypotheses and actually learn something about the disease itself. So I think really emphasizing the use of virtual patients to explore different hypotheses in addition, and the uncertainty in addition to the variability is extremely important.
I would like to add that in terms of exploring quantification of uncertainty and variability, I think in addition to computational method that all the speakers have discussed and presented thoroughly, I would like to bring up the variation at a genomic level and proteomic level, and also the genotype and phenotype association relationship variability between the genotype and phenotype association. And I think those biological variations should be taken into account, will play a very important role in the model credibility in terms of model prediction.
I totally agree with that. And I think that as we are moving forward with these virtual twins, utilizing all this information that may be available from deep profiling these patients will be very, very useful. I mean, there is expression profiles already available that could be implemented into these models, and it could give you a way to capture some of the variability in a very mechanistic manner. So yes.
Yeah, so to follow up on this, and we're talking a lot about the potentially using this for design, and one of the things that we're talking briefly with Dr. Stephan Schmidt earlier about was optimal design. And a question that I have, and we've come up with, do you see opportunities for the use of optimal design for QSP models or systems? And I can certainly. We can start with Dr. Stephan Schmidt, but if anybody else wants to add any experience with optimal design in the QSP space, I would welcome that.
Yeah, so I personally, I think absolutely, and I think that's maybe one of the strong suits also of QSP models that as outlined also by the definition by Van der Graaf and Benson, is that you're trying to understand the dynamics of the system. And trying to understand like key drivers of the system. If you run global sensitivity analysis, then I think you have already a first idea what's driving the system. In other words, what's the rate limiting step, and that can also help biomarker identification. And so then doing optimal design to see when you should take a sample, when this marker is sensitive. I think may shed a lot of light on what's going on under the hood, maybe also provide an opportunity to understand differences between patients, maybe differences in the onset and the progression of the disease. So I think it's could.
Can I add a thought? So, because I think this is kind of related to consensus models, right? If you're gonna, and that came up in our question list, and we're not talking about it. Right, and I think if we're going to build a consensus model, right, we want it to be optimally designed. So, I mean, we've done it, right? There are consensus models to some extent that are commercial in the PBPK space, and in the QSP space, we have I think a very more diverse range of applications. So, right, it's not the same subsystems that we're simulating multiple times and very similar transporter and enzyme considerations, right? So there's going to be a little bit of a challenge of scale, for QSP. But if we think about like optimal design, kind of across, what are the characteristics that we want to see in these consensus models? I mean, I think there has to be some, right? There should be some practical considerations too, in terms of, how these models are actually going to run. So if we have a, cardiovascular model, do we want to really simulate every QRS sort of depolarization, or are we going to kind of abstract these at some time scale to kind of separate out like the renal system from the heartbeat, if we're looking at sort of the some broader renal cardiac inputs, and then maybe, right, we need to have models that basically simulate well, and then some of the more, I think, technically challenging aspects, right, then maybe we have modules that kind of can address simulate this module every so often to get an output, and maybe some design principles like that will enable us to converge on some models that can be more broadly applicable.
Yeah, great. It's great that we're tying it back to the idea of a consensus model. In my mind, this is something that's interesting to me as we start seeing more QSP submissions come in. In some particular areas, we're seeing a similar model being submitted over and over and being used in different applications. And I wonder if just that experience across different customers using the model on different sets of data will build a certain level of validation and verification on its own. But so with that, I think we're at time. I want to thank you all for joining and the great talks today.
Thank you.
Thank you.
Thank you.
Thank you. Take care.